This video will cover subtopic 2.9, photosynthesis. Here are a list of understandings, applications, and skills that will be covered in this video. So, to start with photosynthesis, so it is a metabolic pathway, and this is where carbon dioxide and water get used to produce carbohydrates, and we'll say carbohydrates in the form of glucose, but really it could be any type of carbohydrate. And then at the end of this, or during this process, oxygen gas gets released as a waste product. So the reaction equation shown here gives you the overall reaction that occurs. So you have 6 molecules of carbon dioxide reacting with 12 molecules of water. Light is required in order for this reaction to happen, and this happens on these pigments referred to as chlorophyll. And then very downstream from all of this, you'll generate glucose with 6 oxygen molecules and then 6 water molecules as a product. Now, in photosynthesis, light energy is going to be captured and then transferred to chemical energy, which is stored in the bonds in glucose. So as those carbons get linked together to create glucose, this will ultimately store that chemical energy that gets released or used in cellular respiration. Carbon is going to be fixed from carbon dioxide and used to produce glucose. Water on the reactant side is going to be split with oxygen released as oxygen gas, and then the hydrogens get used to make glucose. Glucose is then used in cellular respiration, can be stored as starch, or used to build cell walls as cellulose. And remember, photosynthesis, in this case we're talking about this occurring in plants, so as they generate that glucose, it will get used to create things like amylose and amylopectin, or cellulose as well. So with photosynthesis, it is harnessing that energy of light. And then just to go through the different types of light, just a reminder that light is made up of all wavelengths of electromagnetic radiation. So there's varying wavelengths of light or electromagnetic radiation. Shorter wavelengths of light will have higher energy. So for example, like X-rays and gamma rays are pretty short wavelengths, and they have high amounts of energy because of those short wavelengths. And then longer wavelengths will have lower energy, such as radio waves or shortwave radio. So they have longer wavelengths and will have lower amounts of energy. Now, found in that electromagnetic spectrum is what's referred to as the visible portion of light. And the visible portion of light is basically that part of that electromagnetic spectrum that we can see with our eyes. So that visible portion is a very tiny portion of that overall spectrum. And this makes up that red light all the way to that indigo and purple light that we can see. And then anything beyond those wavelengths, such as the ultraviolet rays and the infrared rays, are invisible, meaning we don't perceive them, we don't detect them. Other organisms might be able to sense them, but we certainly can't see them. We make use of them in our everyday life. So for example, infrared rays are used in remote controls. So when you change the channel on your TV, you're using infrared light to control that volume. The wavelengths of light that are detected by the eye are also the same wavelengths that are used in photosynthesis. So it is this visible portion of the light spectrum that gets used in photosynthesis. It is this part of light that gets harnessed by chlorophyll. So with that visible light and photosynthesis, it can help start to explain why a leaf will start to look green. So If we have our light coming from the sun, and that light is made up of all wavelengths, but we'll just consider that it's made up of blue, green, and red, and together these would make up white light. As that light travels towards a plant, the plant will absorb the blue light and the red light, and that green light gets reflected from the plant. And that green light that gets reflected is basically what travels to our eyes in the color that we see. And this is why leaves or plants appear to have a green color. And just a reminder of a few things in terms of color theory. You know, a white object does not absorb light, so an object would appear white if it reflected all colors, all visible colors, back to our eye, and we would perceive that as being white. And then a black object absorbs all light. So if a leaf were to appear black, it would absorb the blue, the green, and the red wavelengths of light. One of the things that you'd be expected to do is to be able to draw an absorption spectrum or an action spectrum for photosynthesis. So we'll start with an absorption spectrum. So an absorption spectrum is a graph showing the percentage of light that is absorbed at each wavelength by a pigment involved in photosynthesis. So the x-axis should be labeled as wavelength with nanometers as the units, because this is what wavelength it's measured in. So the scale would range from 400 to 700 nanometers, as these are the wavelengths that correspond to the visible portion of light. The y-axis should be labeled as percent absorption, and because it's percent, the scale should range from 0 to 100. And then data points, if given, for a specific wavelength should be plotted, and then a smooth curve drawn through them. So the following diagram shows what this absorption spectrum should look like. So again, we have the wavelengths of light on the x-axis, with the percent of light absorbed on the y-axis. You do not need to include what color or what wavelength corresponds to what color. This is just down here for reference purposes to indicate, you know, that at around 400 or 450 to 500 nanometers, you know, this corresponds to blue light, and you know, you can see that wavelengths of around 650 to somewhere between 700 correspond to red light. So this is only here for reference purposes. In the absorption spectrum, chlorophyll A and chlorophyll B are plotted. Chlorophyll A is the red line, chlorophyll B is the blue line. Both chlorophyll A and chlorophyll B start to have peaks in that blue spectrum, so starting somewhere at about 425 all the way up to about maybe 475. A lot of light gets absorbed in this range of wavelength by chlorophyll A and chlorophyll B. And then as the wavelength increases into this green portion of light, the absorption drops dramatically. So almost no light is absorbed in this green wavelength. In fact, the majority of this light is going to be reflected. And again, this is why we see a plant as being green. And then as we come into that orange-red end of the light spectrum, absorption increases again for both chlorophyll A and chlorophyll B. So in your drawings, the absorption spectrum for each chlorophyll pigment should cover that range of color. So blue having peaks at the blue end around 450-ish, and a second peak again in that red area of visible light. For an action spectrum, this is a graph showing the rate of photosynthesis at each wavelength. So again, the x-axis should be labeled as wavelength with nanometers as the units. The scale should range from 400 to 700 nanometers. And the y-axis should be used as a measure of the relative amount of photosynthesis. So it could be done as a percentage of maximum rate. So the scale would again range from 0 to 100. And then data points again, if provided, should be plotted, and then a smooth curve drawn through them. So what does this action spectrum look like? So again, you have your wavelength here on the x-axis, and then the rate of photosynthesis on the y-axis. And so what this action spectrum is showing is that, there is a peak somewhere at around like 450, 475 nanometers, which corresponds to that blue light. This means we have a high rate of photosynthesis. The rate of photosynthesis then decreases as the wavelength increases, and then will increase again at around that 650, 675 nanometer range. Corresponding to that red light again. So rate of photosynthesis peaks again at that red light. And one thing you should be able to note are the similarities between an action spectrum and an absorption spectrum. Basically, where you find a peak on the action spectrum corresponds to a peak on the absorption spectrum. So where lots of blue light is being absorbed is where you would expect lots of photosynthesis to happen. And then same with that red wavelength of light. So in photosynthesis, oxygen gas is produced. And oxygen gas is produced from a process referred to as the photolysis of water. So in the following reaction, which you have on your notes, as that water reacts in photolysis, so here we have water, and you might want to consider putting this reaction down on your notes in the space provided. So with two molecules of H2O, you'll have light that gets absorbed, and the lightning bolt is supposed to represent light. The water will get lysed to release the 4 hydrogen ions plus 4 electrons, and then you have 1 molecule of oxygen gas that gets created as a byproduct from this photolysis. And this is what gets released. The hydrogens are used to create glucose, and then the electrons get used in the light-independent set of reactions, or the remainder of the light-independent reactions. So all of the oxygen generated from photosynthesis will come from the photolysis of water. This reaction will only occur in the light, as you do need light to lyse the water. And then oxygen that is produced as that waste product diffuses away. So in the picture down here, it shows the reactants as 6 CO2 and 12 H2O, and then the products, the glucose, the water that is generated, and the oxygen gas. And then the arrows just show where each of the atoms are going into the different products. So obviously all of the carbon from CO2 goes into the carbon in glucose. The oxygen from CO2, some of it will go towards the oxygen in glucose, some of it towards the oxygen in water. From the water, all of the hydrogens are used to create glucose, and then some of those hydrogens are used to create water, and then all of the oxygen gets used to create oxygen gas. So now, in that process of photosynthesis, as this has been happening for millions of years, or billions of years, this has had an effect on Earth's atmosphere, oceans, rock, and rock composition. So primordial Earth, that very early Earth, had a reducing atmosphere that contained very low levels of oxygen gas, approximately 2% of the atmosphere was oxygen gas. Now, somewhere about 2.5 billion years ago, a type of bacteria called cyanobacteria containing chlorophyll started performing the first photosynthesis reactions. And those photosynthesis reactions started creating oxygen gas as a byproduct. So as that oxygen gas got produced, that oxygen gas started reacting with compounds in the oceans, and they started reacting with metallic compounds, specifically like iron compounds in the ocean. Those iron compounds were oxidized, creating iron ore, iron oxide, for example, which precipitated onto the seabed. And it's that iron ore that we go and mine and obtain like our iron ore from, which we then purify iron. Once all that iron had in the oceans had reacted, then that oxygen gas started being released out into the atmosphere, where those oxygen levels rose to about 20% of our atmosphere. So for about 750 million years, our oxygen levels have been increasing to about 20%, where they remain about stable. So our atmosphere is about 20% oxygen gas. Now, oxygen gas going out into that atmosphere also allowed the formation of an ozone layer. Get rid of that X. That O3, which helps protect Earth from high levels of UV radiation. There's lots of radiation out in space, especially coming from the sun. That ozone layer absorbs a lot of that UV radiation and protects us from a lot of that radiation. Now, energy is needed to be able to take carbon dioxide and turn it into a carbohydrate. So as shown in the reaction here, like light is needed in this process. And so this process or this reaction is referred to as an endothermic reaction, meaning it requires an input of energy, and that input of energy comes in the form of light. So the energy is needed for the conversion of carbon dioxide into that carbohydrate, and this is obtained by absorbing light. So that process of photosynthesis can only occur in the light. And then at the end of it, when glucose is made, that glucose can be converted into polysaccharides, such as starch or cellulose. Now, there are factors that do affect the rate of photosynthesis. So, for example, light intensity is one factor that will affect the rate of photosynthesis. So at high levels of light intensity, further increases in light have no effect on the rate of photosynthesis. So if we have a look at this graph here, so we have our light intensity on our x-axis, our rate of photosynthesis on our y-axis. And then at low levels of light intensity, there are low levels of photosynthesis, or the rate of photosynthesis is rather low. But as that light intensity starts to increase, that rate of photosynthesis will start to increase as well. And so they'll reach a point where a plateau is hit, where the rate of photosynthesis no longer increases. And any increases in light intensity will not have an effect on that rate of photosynthesis. So at this point, light intensity is no longer a limiting factor, and other factors such as temperature, CO2 concentration, or enzymes, or chloroplasts working at a maximum efficiency is now limiting photosynthesis. Another factor affecting the rate of photosynthesis is CO2 concentration. So again, on our x-axis, we have CO2 concentration increasing. We have rate of photosynthesis on our y-axis. And so at low levels of CO2, there is a low rate of photosynthesis. But then as carbon dioxide concentrations increase, rate of photosynthesis will also increase. And so eventually there will come a point, a certain point of carbon dioxide concentration, where that rate no longer increases, and this is where we've hit a plateau. So above this carbon dioxide concentration point, so above this point, any further increases in carbon dioxide concentration will not affect the rate of photosynthesis. And then at this point, CO2 is no longer a limiting factor. The last factor we'll look at affecting the rate of photosynthesis is temperature. So again, we have temperature on our x-axis and rate of photosynthesis on our y-axis. At low temperatures, there is a low rate of photosynthesis, but as temperature increases, this will give the molecules more kinetic energy, causing substrates to collide with active sites more frequently, and this will increase the rate of photosynthesis. So photosynthesis will start to increase until we reach a point where we reach a maximum rate of photosynthesis. And this will occur at an optimum temperature. So at some temperature, we will hit a peak. And as the temperature approaches the optimum, the enzymes will begin to denature. So those enzymes involved in photosynthesis will begin to denature, causing the rate of photosynthesis to increase more slowly and eventually peak. And then after the optimum temperature, there is a very rapid drop in the rate of photosynthesis. And basically above that optimum temperature, enzymes will denature a lot more rapidly, causing a fast decrease in the rate of photosynthesis. So as part of the nature of science for this subtopic, this is dealing with an experimental design, controlling relevant variables in photosynthesis. In these photosynthesis experiments is essential. So there are some questions that you need to answer when designing an experiment to investigate a limiting factor on photosynthesis. So you have to determine which limiting factor will you investigate. This will become your independent variable, so this will become the variable which you manipulate. And because you're investigating photosynthesis, you have to determine how will you measure the rate of photosynthesis. So this will be the dependent variable. And then you have to consider then how will you keep other limiting factors at a constant and optimal level. And these are the control variables. So photosynthesis, when you're thinking about your dependent variable, photosynthesis can be measured in various ways. So for example, you can measure carbon dioxide uptake. In things like aquatic plants, you can measure a change in pH of the water as CO2 is removed from that water. For terrestrial plants, you could use a carbon dioxide gas sensor to see how much CO2 is being taken up by the plant. You could also measure oxygen gas production. So in aquatic plants, this is basically counting the number of bubbles produced from that aquatic plant as it performs photosynthesis. You could also place a graduated cylinder over the plant and record the volume of water that has been displaced. For terrestrial plants, you could use an oxygen gas sensor to measure the quantity of oxygen that has been produced. You can also measure a change in biomass. So as photosynthesis is performed and the plant is generating carbohydrates in the form of starch and cellulose, this will allow the plant to grow and increase in mass. So you could also measure the dry weight of the plant as a measure of photosynthesis as well. So this is the end of the video for subtopic 2.9 on photosynthesis.